This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 36 for June 12th, 2009. Hi, everybody. I'm Vincent Racaniello, and we're recording in New York City today. And joining me, in no particular order, is Dick Despommier. Welcome, Dick.
Thank you, Vince. Pleasure to be here again.
Dick's in my office, as is Hamish Young. Welcome back, Hamish.
Good afternoon, everybody.
And from Western Massachusetts is Alan Dove.
Here I am, yes, via Skype.
So we have 4 people today to talk about viruses. And of course, the big news is it's officially a pandemic.
Right. It's officially a pandemic.
Right. Oh, I think I had heard that mentioned somewhere.
I'm about to take back everything I ever said about this.
I think Vince and I were right. I think we said yes.
Okay, it is much ado about nothing. It's much ado about something now.
Oh, Dick, people are dying, right?
Well, 144 people did die.
We don't want to make light of this. But
of course not.
I do remember you saying this was going to fizzle out, and Hamish and I thought when we last talked about it together that it would be a pandemic. Peter Palese took the safe road. He said it's a 50-50 chance of being a pandemic.
That's not fair.
Right? Yeah.
And
as soon as I saw it in Australia,
you said,
that's it, because there were like 10, and then all of a sudden there were 100, and then there was last time I looked there were 1,400 or 1,500 or something. So
and Chile too.
Chile, yes. Well, all the southern hemisphere. And I said, that's it.
But there's not a lot of people that live in the southern hemisphere, so
Chile, Santiago's a pretty big city.
Yeah, but compared to the world population, I mean.
Yeah, true.
Well, we didn't need WHO to tell us it was a pandemic.
I guess
they just made the official announcement. They raised the alert to level 6, pandemic alert. Right. But we probably knew it was coming for a while. If you look at the numbers, and now WHO has a very nice chart or graphic, if you will, on their website. Let's see if I can find it here. Here we go. They have a map where now they're putting red dots all over the globe, and the size of the dot can tell you how many cumulative cases there have been.
So it's like a plaque assay.
Yep. Particularly an adenovirus plaque assay, because the plaques are different sizes.
Very good. They range from 1 to 10, 11 to 50, 51 to 500, and then 500 or more.
Right.
So you can see that South America now has a lot of large dots. Australia. But Africa is conspicuously void of dots except for Egypt, which I suppose is a surveillance issue.
South Africa has very good surveillance.
Nigeria should be reporting in.
I read on some websites that there are cases in South Africa. I don't know why they're not showing
There's another nice graphic on the BBC where they show you draw a little arrow along the bottom of the map, and it shows the increase in numbers over time. It's based on the WHO numbers.
Oh, that's very nice.
And so you can see it quite graphically going from March, when it started in Mexico.
Well, we are getting better at quantitatively expressing our results.
Right.
So where would I find it? At the BBC homepage?
Yes. And look at any article which is dealing with swine flu, and you'll, related links.
Okay, let's see. Here we go. Country by country. Is that? Here's the slider. Move the slider to see how the disease has spread.
Right. So I thought that was quite a useful.
Oh, that's very nice.
Yeah.
So you can go all the way back to.
So it's like almost like a movie.
21st April, exactly. It's like a quick time movie.
Right.
And now we see United States, Canada, Mexico, Europe. Right. And then eventually, and the circles get bigger. That's great. We'll put a link to that.
Yeah, I thought that was a good one.
Very nice. Thank you, Hamish. You are keeping up with viruses, I see.
Well, certainly with this, because I think this is one of the most interesting things to have come along in a while.
So globally, we have 29,000 cases, almost 30,000 cases, 145 deaths.
Right.
Let's see what the the case fatality ratio is. 145 divided by 30,000. You could probably do this in your head. 0.5%.
Right.
A little higher than typical, but not as high as the initial Mexico numbers suggested. Right. So it's typical.
But the Mexico numbers have come down, haven't they? The numerator's gone up and the denominator's gone down. So
it's about 0.5% in Mexico as well. And so the spread, as you say, is what really tipped us off that it would be a pandemic.
Right.
Well, and do we have any more data on the age distribution of the deaths?
Probably.
Mm-hmm.
anecdotally from the newspapers, it says that it's still predominantly in the young and those under 50.
Okay.
Because,
or with other underlying medical problems.
Well, the deaths are said to be, but of course, as somebody pointed out, the number of things that are classified as being underlying medical problems, about 50% of the New York City population has one or other of those things.
Right, exactly.
So it's pretty widespread.
I once heard a doctor comment that the definition of a healthy person is someone who hasn't been sufficiently tested yet.
Boy,
that's a doctor that wants to make a lot of money, I'd say.
That reminds me of the doctor's dilemma and stimulate the phagocytes. Do you remember that?
The doctor's dilemma.
We're reliving it daily.
I don't know. I don't have any numbers. I just looked for age distribution, but I'm not sure CDC has that up yet. But,
so now, if this had occurred in January, February, and March, would anyone have been surprised?
Surprised in what sense?
Of this flu epidemic.
Well, it would have been a new emergence.
That would have been a typical reveal for this virus to have spread in this fashion during the flu season.
Yeah, but pandemics often come up towards the tail end of flu seasons.
Right.
Pandemic strains. It's typical. So but we'd be just as surprised as we are now. People were surprised that it was a swine virus. Everyone was suspecting an avian virus.
That's right.
So in that sense, I am surprised that it's still continuing in the U.S.
Yes, I mean that's the thing that I think that is surprising people is the prevalence.
So if you look at the CDC graph by week, so at the end of week 15, this flu season looked almost over, and suddenly we had this huge peak, and now at week 17, 18, it was as high as in terms of isolation as before, and it started to go down, but now it's plateaued. Week 19, 20, and 21, the number of isolates are the same.
Yeah.
Mm-hmm.
And the percent positive specimens are still rising, actually. Now, the CDC...
Now, is that all flu, or is that...
This is all flu. The dark blue bars are the novel H1, but they're still getting H3N2 in the previous H1N1, but not very much. In fact, on this last bar on week 21, there is no more of the old H1N1.
Right. And I guess we should be clear in that there is no H5N1 in any of this.
No H5 in any of this, although there are still some cases here and there.
That's true. I think Egypt.
Egypt is the case.
Yeah, that's
the
right.
worry.
Egypt
is the worry.
Right.
Now, CDC says during week 21, influenza activity decreased in the U.S. However, there are still higher levels of flu-like illness than is normal for this time of year.
Right.
And I heard something in the Times yesterday saying that New York City may have, in fact, more cases than we know about. There may be as many as half a million or something like that. And I also have, let's see if I can find this. Yesterday, my school district sent out, I live in a little town in New Jersey, and the school district sent out a letter saying, we feel it is important to alert you that within the last week, we have seen an increase in student absences due to flu, bronchitis, and strep. While we have not had a confirmed case of swine flu, we are continuing to monitor the reason for students' absences. Therefore, we are instituting the CDC control guidelines, which means anybody with influenza-like illness should stay home for at least 7 days. So that's interesting. Apparently, it is still continuing, and I just wonder if what has to do, because we think that it's seasonal. It should be stopping by now in the Northern Hemisphere. I wonder if our surveillance is intensified, and that's why we're picking it up, whether the weather has been abnormally cool
and moist,
and that has something to do with it, or whether the lack of immunity transcends all the determinants of transmission.
But Vince, when you just said about weather, I mean, in so many other places, they've had so many different kinds of weather that you can't really pinpoint
I'm
weather.
just basing it on my personal experience, which is flawed. I understand. You're right. You're absolutely
Seriously
right.
flawed in that sense.
Absolutely, it's a valid point. It's a valid point where you're looking at things like the New York City case incidents. What's the weather been like in New York City? And the Northeast has been kind of in a cool spring.
Yeah, but you look at Mexico or Australia or places like that, and the weather patterns are totally different. So
and actually,
I think they're going to throw out this hypothesis actually about weather-related transmission cycle. I really do, based on its pandemicity, because the weather's got to be different every place.
So, what do we think, gentlemen? Will it continue to circulate through the summer in the Northern Hemisphere?
I'm staying out of this conversation. I'm out of it.
I'm going to bet on the opposite of whatever Dick said.
Okay, I think it'll continue.
Very good. Take the opposite.
As a total ignorant person in this regard.
Well, you see, the problem is the surveillance is very intense now, so we might not have picked this up normally. And I would like to see in a typical seasonal flu non-season with intense surveillance in the future. We'll have to do that, what you would see. But what do you think, Hamish?
I think the surveillance issue is probably correct, because one of the things that WHO has been saying is that the scare about H5N1 has made countries much more proactive than they would have been. And so I suspect that there is a greater surveillance. For example, I follow fairly closely what's happening in Scotland, being from Scotland, and they are taking a very active role in following all of these instances. And transmission is still going along. Scotland, even though it's got about a tenth of the population of Britain, has about a quarter of the cases, which were initially brought in from Mexico and are spreading through primary schools and secondary schools. And they are taking they seem to be testing everyone. So you wouldn't normally do that in a flu season, it seems to me. So that's a microcosm, I think, of what's going on worldwide. So I suspect that surveillance is playing a role, which I think is good. I think we'll get lots of useful information, which can be used, and I think is being used to try to mitigate the effects of this.
Also, if the surveillance is done at the right resolution, in other words, if these samples are really being tested to see whether it's one of the regular circulating strains or H1N1, then that'll provide extremely usul information, because we would be able to see how long the regular strains circulate in the regular season.
Yeah, no, that's very interesting.
Yep.
We should point out that, in fact, in the textbooks, it says in the Northern Hemisphere, in the summer, you can always isolate influenza virus from large cities. It doesn't actually go away. So this may be correct that if with higher surveillance you see it all the time. But the key will be whether there are outbreaks of thousands of cases here and there or not during the summer. I would be surprised to see that, but we'll see. We'll keep you posted, because TWiV will go over the summer. We don't take a summer break, except for Dick, who's going to Qatar.
That's
Qatar.
Cotter. Welcome back.
Welcome back, Cotter.
That's right.
And
is summer really the season to go to Qatar in, or does it matter?
It's probably all one blur, but it really does get intensively hot over there in the summer. There's no question about it. I'll let you know.
Yeah, Dick is going away for a few weeks, so we won't have him to bother.
That's right,
and
or to
annoy.
make fun of.
And in fact, you may leave shortly today because you're busy. But
I'm afraid I have to.
You have a half hour. You can't leave yet.
I'm not leaving.
All right.
I'm stuck
in
in my
fact,
chair here.
What this means, among other things, is that in the fall we should probably get a flu vaccine against the new strain.
Yeah.
Will you get it, Hamish?
Yes, I'll certainly get it.
And whether we still need the old one or not depends on what circulates, I think, in the Southern Hemisphere, what they isolate. So we'll keep an eye on that. And Alan has a story about potential new flu vaccine, right?
Yes. So I just rearranged the agenda a little bit. I'm glad you noticed. So it looks like Novartis has produced the first batch of vaccine against H1N1. And this was surprising to me that this has already happened, because they just distributed the reference strains a couple of weeks ago, if I'm read that correctly.
That's correct, yeah. I think we talked about it just last week on TWiV. Yeah, 2 weeks ago, yeah.
Right, and listeners should bear in mind that we're recording a little bit in advance for some of these episodes. But yeah, Novartis has just announced they've produced a batch of H1N1 vaccine, and they did it in cell culture, which was the other interesting thing. So they're using a new production approach. As many listeners probably already know, as we've talked about, eggs are one of the main problems in growing up large quantities of flu vaccine, because you have to culture it in eggs, you have to get an egg-adapted strain and all that. So just today, when we're recording, June 12th, Novartis announced that they've got this test batch. They're going to use that for preclinical evaluation and probably an initial round of clinical trials. And they are gearing up. They claim that they're going to be able to produce millions of doses of vaccine a week. So obviously their cell culture techniques are a lot better than mine ever were.
Are they also using the proprietary adjuvants?
I think.
Wait, was that Novartis?
No, that was Glaxo.
No, that was Glaxo.
Maybe they'll collaborate then.
So is this production going to be in cell culture as well as the production as the original?
I think so, because the This batch that they produced, they say well, according to the Associated Press, they produced it at a Novartis plant in Marburg, Germany, of all places.
Oh, isn't that wonderful? Yes.
We've got a chance for 2 here now. Come on, let's go for it.
So maybe they could title it the Marburg vaccine.
It's ironic that you should mention that because the year that outbreak occurred, I had the privilege of visiting Höchst, which is where the Marburg virus was released and killed 13 people.
Yeah.
So they were acutely aware of the introduction of wild virus strains from various places.
And
I think they've overcome that.
Novartis is in the process of building another plant in North Carolina. To also be vaccine production, presumably with cell culture.
So is this cell culture vaccine licensed in the U.S., Alan? Do you know?
I not that I'm aware of, but that doesn't mean much.
It can be licensed by the fall.
Yeah, they could certainly get it licensed under the current conditions, I'm sure.
Yep. Or compassionate use.
Yeah, and in any case, in general, the FDA has taken a fairly favorable attitude toward vaccines, where you've come up with a cleaner production system. And I think it'd be fairly easy to argue that a cell culture production system is cleaner than an egg-based production system.
I wonder what cells these are grown in. Does anyone know? Probably a diploid fibroblast like WI38 or something like that?
When we used to do it for medical students, when we were growing up stocks, we would use MDCK cells, I think, which is a canine kidney.
But they're transformed. They wouldn't be licensed to do this.
Maiden Derby canine kidney? No, but are they transformed?
Yeah. Huh. I believe MRC5 is one line.
I thought there were a couple of transformed lines that were approved for at least protein drug production. I don't know if they're approved for vaccine production. I could be wrong.
Not live vaccines. No.
Okay.
Or infectious vaccines, we should say.
Right.
My perusal here is not yielding what kind of cells they're grown in.
No.
Now, if we were doing a live version of this show...
You might want to try going to ProMed, Vince. Maybe it's on ProMed.
Well, we won't waste your time because I know you have to go. But if we were doing a live version, we would have people telling us right now in real time.
Most likely.
Maybe that's the program for next time.
So killed vaccines, presumably this is a killed vaccine.
This one is
Novartis.
I think it will be killed.
So is there a restriction on cell culture type when you use a killed?
Yeah.
Really?
Because, the DNA in there is still potentially transforming. Yeah. So the polio vaccine, the killed vaccine was grown for many years in monkey kidney cells.
Right.
But I think in Europe they use MRC5s.
Yes, they do. Yes.
Which is a more normal kind of cell line.
It's a diploid fibroblast.
Yeah. So I'll look it up.
It's the European equivalent of WI38.
Right. So this vaccine could be produced earlier than the egg-grown vaccine, I think.
Yeah, they're claiming production capacity in the millions of doses of vaccine a week, which is pretty impressive.
Yeah,
it's a lot. Yeah, great.
So they could be, business-wise, they could be first to market with this, and that would be great for them.
Sure. Well, look for it at your local airport.
Yes.
Really, I was at O'Hare Airport about 6 months ago, and they said, "Come over here and get your free flu vaccine." So
shouldn't they
coming soon to a hypodermic needle near you?
That's right.
Shouldn't they give it away?
That's exactly right, shouldn't they?
Why should anyone have to pay to stay healthy?
Well,
this is a question we can't go into.
We've got 10 more shows lined up.
I think we should direct you to the AMA, Vince.
All right. Doctors. Well, I'm not saying that doctors don't have to charge, but all right, we'll save it for another show. All right, we have a couple of 2 more non-flu stories to go on before we get to our main topic. But before we move on, here's a message from our friends at Citrix, the makers of GoToMyPC.com. And as all of you know, I rely on my office computer. I would be lost without it, and I would be lost without GoToMyPC. I can access my computer from anywhere as long as it's on the internet, and that way I can work from anywhere. I'm not stuck at the office. And GoToMyPC is the only remote access service that I recommend. In fact, I just used it last week. They have another product called GoToMeeting, and I used it to interact with someone. You'll love accessing your office PC from home. When there's traffic, you can stay home. Very easy. Log on to GoToMyPC.com, and your PC desktop instantly appears. You can use any program, work on any file, check email, and access your network resources. I was just at a party a couple of weeks ago for my son's lacrosse team. I was telling his coach, who is a lawyer, about TWiV. He said, "How do you make money?" I said, "Well, we have a little support from Go to My PC."
That was his first question?
Yeah. And he said, "I use that.
Tell
It's
him we're not
great."
interested.
He said, "I use Go to My PC. It's a great program. I use it all the time." You can try Go to My PC free for 30 days, but you must visit gotomypc.com/podcast. That's gotomypc.com/podcast, all one word, for a free 30-day trial. We thank them for their support.
When I was growing up, Vince, I had when I was younger, we had another program that I responded to. It was called Go to Your Room.
And who made that? Was that Citrix?
No, actually it was an earlier manufacturer.
Despommier Senior.
It was a maternal organization.
Now, Dick, here's a story from Alan, which I think you will like very much.
Okay.
Midge keeps invasive mosquito in check, aiding native mosquitoes. What's that all about, Alan?
Yes, so this came up in the press releases that I get and really caught my attention. So as Dick is aware, and I'm sure many listeners are as well, there's an invasive mosquito, Aedes albopictus, which is responsible for carrying quite a few diseases, including dengue fever, to pick one example.
Whose common name, by the way, is the Asian tiger mosquito.
Yes, it is known as the Asian tiger mosquito. This is one of the many fascinating organisms we've imported from Asia.
That's right.
Of course, we've done the same to them, sent things their way. But so the problem, of course, is you've got these mosquitoes that can carry this virus, and they're breeding now in the United States. And the thinking has been that one of the reasons they're so successful, besides being mosquitoes, is that they don't have a lot of natural predators in this environment that would keep them in check. Of course, they're also quite common in Asia, so even in the presence of natural predators, they're
that's right.
a fairly successful mosquito.
Yep.
So this work just came out earlier, I guess, last week, and it's researchers at the Illinois Natural History Survey did a sort of a co-culturing experiment in the lab where they had looked around and they found that the Asian tiger mosquito is often breeding in the same sorts of environments that the eastern treehole mosquito, Aedes triceriatus, breeds in. These are both container breeders, they're called. Right. So
all of the Aedes are.
Right. So any little puddle on the road, the treehole mosquito, a hole in a tree.
Tin cans.
Tin cans, dropped coffee cups, that sort of thing.
Stored tires. Right.
Yep. So they found, interestingly, when you sort of recreate the little ecosystem of these puddles in the lab, and you add the larva of a midge, Corythrella appendiculata. It's just a midge that the larva breeds in these same sorts of puddles. Right. It gobbles up mosquito larvae, as is its habit. It's one of the nice things. I mean, it's a little fly. It's a little midge. So
it's a predator species then.
And it's a predator. The larva is a predator
species.
Yeah,
I like that.
Mosquito larvae.
Yep.
The interesting thing is that this midge seems to have a preference for Asian tiger mosquito larvae.
Now, how can that be? It's only been here for the last 15 years.
Well, the reason is that the tiger mosquito larvae are smaller.
Oh, I see.
And the midge is larva is a certain size, and the native mosquitoes, probably because they've had this war going on for quite some time, their larvae grow to a larger size that's harder for the midge to catch and get a hold of.
Got it.
So there's a skewing of the population. The midge tends to preferentially prey on the invasive species, which is, of course, what you'd really like to see.
You bet.
And, the usual caveats apply. This is a laboratory study. We don't know if this is actually going on in nature.
Right.
But it's certainly a promising start.
Right.
Did we discuss how the Asian tiger mosquito actually got into this country?
We did.
Fine, then we don't have to cover that anymore.
In tires, right?
Yep, from Japan.
Yes. Right.
Recycled tires.
The good thing about this study, it seems to me, I haven't read it, but from what your description, is that the predator is native to this country.
Right.
Yes.
Whereas previous attempts to introduce predators to take care of an invasive species have often been fraught.
Yes.
So it seems to me that if this is true, then we're going to evolve a nice ecological balance. We're not going to get rid of Aedes albopictus, but
it'll keep down the population.
It'll keep down the population.
One of the interesting things also to note, though, however, is that the Asian tiger mosquito is a not so efficient vector of yellow fever, whereas the Aedes aegypti mosquito is, and this mosquito outcompetes Aedes aegypti for breeding sites. So there is some trade-off in favor of keeping
I'd rather get dengue than yellow fever, is what you're saying, Dick.
That's right. It also transmits lacrosse virus, and it transmits
which does albopictus.
Albopictus, yes, it does.
Lacrosse is not
as well.
nice. Yeah.
So how would you take advantage of this? Would you seed midges all over, or how would you do that?
You could.
You could.
I think the real take-home lesson from this is that you probably want to avoid using broad-spectrum pesticides when you can.
Right.
Because this is certainly a problem with control of many insect species.
Sure.
Fire ants come to mind. If you go through and you wipe out all of the arthropods in an area, initially results are great because you don't have any more mosquitoes.
Right.
But in many cases, the invasive species will recolonize first and end up alone in the environment before the predators come back. And so this adds to a cautionary tale that you don't necessarily want to wipe out all the flies. You just want to see if you can go in and target them. It probably wouldn't hurt to go introducing midges. As Hamish pointed out, this is a native species.
Is it all over the U.S., do you know?
It can't be all over the U.S., but it's called the eastern treehole mosquito. My guess is that it is it.
Midge.
I'm sorry. It's the midge is
probably doesn't have a common name.
It's not a biting midge either,
no,
presumably.
I don't think it's a biting midge.
This would be tragic if it was, and we were to go ahead and introduce it. It turns out it can't transmit. No, we won't go there. But there's another predator species that we should mention at the same time, and that's another mosquito. It's called Toxarin chaetini, and it's huge. It's actually triple the size of normal mosquitoes, and it doesn't suck blood. So it's a predator species for other mosquito larvae. And it's around, so it has achieved balance already. The difficulty of introducing even an indigenous species as a control measure is that you're never sure of the balance that you'll achieve or how often you have to do it.
Right.
So you could commit a lot of resources to this and still not get the desired result.
While we're on the subject of mosquitoes, what has happened with attempts to introduce large numbers of irradiated male mosquitoes or whatever?
The sterile insect technique. One of my absolute favorite.
Yeah, me too.
So what has happened with all of that? I mean, I remember that
we successfully eradicated screwworm.
That's true. That is true. And it was done just that way too.
Yeah. That was a terrible parasite of cattle in the United States.
It does keep coming back, though, because we can't spray that technology into Mexico. So the screwworm does keep coming back from Mexico, but we've eliminated it from Florida, which is an isolated region. Male mosquitoes produce a substance in their accessory glands. I've actually done some work on this a long, long time ago, which they inject into the female during insemination, which actually closes down the micropyle, which allows the sperm to go between the seminal receptacle of the female and the ovaries. So this material, which we called matron, M-A-T-R-O-N, was turned out to be a very high molecular weight protein of something like 300,000 as we tried to work it out to see whether we could exploit that system or not. If you could get sterile males to mate with normal females before they mated with other males, then they would be sterile. Because they will mate sequentially. Female mosquitoes will mate sequentially. So you can find mixed genes, whereas some other flies like Drosophila, I think they only mate once and that's it. So I don't know what the progress has been on that.
I haven't seen anything about it.
The push right now that I've seen is to genetically modify susceptibility of known vector species to big diseases like malaria. And Tony James, operating out at San Diego State University, who happens to be one of the few medical entomologists, it's also in the National Academy of Sciences, by the way, as a young guy, discovered how to transfect mosquitoes by using Drosophila promoter sequences. That was the only thing stopping that whole process from going forward. So now you can start to select for genes of refractory strains of Anopheline mosquitoes, find out what the genetics is, pick those genes, transfect them into normal strains, release the hell out of it, and try to replace them with these new genes. But, it's very difficult because you have to throw in some crossovers to make sure you can tell which was which to begin with and see how the crossover is transmitted to. And it gets very sloppy. And they're doing some research, though, in Mali, in the country of Mali, through a central research laboratory in Rome. By a guy by the name of Colucci, and Bob Guad's a friend of mine who works down at NIH. You know Bob. Yes, I met him. Bob is a medical entomologist and has been at the forefront of malaria research for a long time. So that's about all I know up to date for mosquito work.
We actually have an email that's about this, so I'm going to read it.
Oh, great.
This is from Ben. Ben writes, in, I believe, episode 10 or 11, there was discussion about the discovery of an RNA mechanism by which mosquitoes avoid developing diseases from the viruses which they carry. This is siRNAs that degrade viruses. A comment was made about how it isn't clear how this discovery could be used to render the wild population of mosquitoes vulnerable, because evolution would select against any artificially introduced mutation in this gene, and we can't outsmart evolution. So he sent us an article from Science. A synthetic maternal effect selfish genetic element drives population replacement in Drosophila. When I first read the article, I wondered if this could be given a payload that included some sort of molecular transgenerational clock that would turn on a lethal gene only after several generations, allowing time for it to be spread throughout the population via the technique described. I had in mind something like the telomere clock responsible for Hayflick limits, only without the reset mechanism. However, your point about an intentional mutation increasing their disease susceptibility might be a more realistic, less controversial, and yet widely applicable approach. So in this paper, the problem is, and they state it beautifully in the abstract, one proposed strategy for controlling the transmission of insect-borne pathogens uses a drive mechanism to ensure the rapid spread of transgenes, conferring disease refractoriness throughout wild populations. So they say the problem is, though, if these mosquitoes that you engineer don't propagate at a greater than 1 Mendelian ratio, they will never take over.
No, that's right.
So they say you have to include something to drive these genes through the wild population at greater than Mendelian frequencies. So what they do here is they make a cassette. They have a toxin that's expressed early in development, which kills the embryo in the mosquito, and then they have an antitoxin, and they put them in together. So they must propagate together. And then if you want to attach a gene to it,
that's a great idea.
You use that in that mosquito strain. And they don't recombine or separate.
Right.
And they show that the frequencies are greater than Mendelian in the propagation of this kind of a cassette.
Very cool.
So you put a toxin and an antitoxin together with in your strain, and it will go through the population. So that's beautiful. And this is with Drosophila, of course, because you can do these manipulations.
Sure.
I presume you can do it in mosquitoes as well.
You can.
Do they have a drive? Is there a genetic drive?
A meiotic
mechanism?
drive, it's called.
Meiotic drive.
That's right. That's right.
Mosquitoes, Drosophila.
Yep.
That's a good question. This gene that they're using is called Medea. Maternal effect dominant embryonic arrest. Here we go. Yeah, they don't say anything about mosquitoes.
Their intention, of course, is to apply this to mosquitoes, I'm sure.
I'm sure they will. I don't know of any.
And who are the authors?
The laboratory is Bruce Hay at Caltech.
Not too far from Tony James. Maybe they're thinking of collaborating.
It's actually Caltech and UCLA.
Okay.
Bruce Hay. Anyway, we'll put a link to that. That's pretty cool.
Yep. Oh, one last footnote. If you're asking for books of the month or books of the year, the original treatise on Aedes aegypti was written by a man by the name of Christopher. And in this original book, which was very thick, it had a single sentence that said, "There are no mutants of Aedes aegypti." Dr. George Craig at the University of Notre Dame, who had studied with Kitzmiller working with Anopheline mosquitoes, then switched immediately to Aedes aegypti and immediately put his undergraduate students to work looking for mutants and found tons of them, of course. So the next thing you know, by the way, it took Morgan and those other people downtown at our own university 3 years to find a single mutant of Drosophila, right? So it's a good point to make. There's a new edition of Christopher's book And it's twice as thick, and it's all filled with genetics.
Right.
So it's come full circle,
Right,
you know.
right.
It's self-fulfilling. Science is self-fulling.
Right.
And
Self-correcting.
self-correcting.
And self-fulfilling. We've filled in the missing blanks. I guess that's what I meant to say.
Yeah.
All right, our last story is very, very briefly, 2 cases of vaccine-associated polio have been identified in India. And I think this is interesting because vaccine-associated polio is polio caused by the infectious polio vaccine, which is used in India and much of the world for eradication. And in the U.S., we had about 8 to 10 cases a year of VAP, as we called it. And that's why we stopped using the live vaccine. Once we eradicated polio from the U.S., there was no longer any reason to have polio caused by the vaccine. Some would argue there was never any reason to have it, but that's a story for another time.
Day, right.
And so India now is getting low on polio, not eradicated, but it's getting low. And here now we're starting to see vaccine-associated polio. So once the distraction of wild polio is gone or low, you start to see it because it's hard to pick up the vaccine-associated cases in a background of wild polio.
So
But now we have 2.
remember, Vince, when I got back from my trip from India, I told you that I saw all these recorded dates on the sides of the walls of the houses as to when they had visited and who got the vaccine. So it's really pervasive.
So I think this will begin to increase. I'm curious to see if the rate will be the same as it was in the U.S., about 1 case per 1.5 million doses, or whether it's any different based on population. So it's an interesting problem because some have claimed that there are some people who sue the manufacturers in the U.S. claiming they made a defective product, but and this is a different manufacturer in India, of course, and if the rate is the same everywhere, it's not the product, obviously, it's the host, but we'll see.
Yep.
Okay, before we move on, let's have a word from our friends at ASM.
Now available from ASM Press at eStore.asm.org, the new book by author Irwin W. Sherman, The Elusive Malaria Vaccine, Miracle or Mirage. Chronicling a 100-year quest, this book tells the fascinating story of the hunt for the still elusive malaria vaccine. Its clear style makes the book accessible to a general audience and brings to life all the drama of the hunt, celebrating the triumphs and documenting the failures. The author engagingly captures the controversies, the missteps, wars of words, stolen ideas, and clashes of ego as researchers around the world compete to develop the first successful malaria vaccine. To view the table of contents for the elusive malaria vaccine, Miracle or Mirage, visit ASM Press at eStore.asm.org.
Now, Hamish, we had a listener ask that we talk a bit about adenoviruses. Mm-hmm. And you used to work on adenoviruses.
Yes, I did.
I remember.
Yes, I did. Yes.
So maybe we could have a little chat about adenoviruses. Okay. What is an adeno? Why is it called adenovirus?
Well, because originally it was isolated from the adenoidal tissues. They were propagated, or cell cultures were taken out of adenoids and propagated. And they couldn't understand why the tissue culture was breaking down and dying. Yeah. And I think that was Wally Rowe. I get my brain
Bob
cells.
Bob Janik, maybe?
No, it was Wally Rowe.
Wally Rowe. What year was this, roughly?
In the late 40s, early 50s. Mm-hmm. I think 53, if I remember correctly. I mean, I'm being pushed. I should have done my homework before I came. But it was simultaneously discovered by the big vaccine man whose name is escaping me.
Maurice
Hilleman.
1953.
See, I even knew that part.
Right. Thank you very much. Vince is giving me a cheat sheet. So
I know that year very well.
Yeah, 53.
I was
born then. Right? So it was simultaneously discovered, and there was a lot of interest in what was called acute respiratory disease of recruits. And our former chairman, Harry Ginsberg, was one of the people involved in this early work because he had a contract from the Army to work out why recruits, in the days of the draft, when everybody was drafted, why they would get together and almost within weeks they would have big outbreaks of serious upper respiratory tract disease. So it was called acute respiratory disease of recruits, or ARD. Well, it turned out that was caused by adenovirus. So there was a sort of confluence of events which led to the discovery of this virus. It's fairly rapidly discovered to be a DNA-containing virus, fairly stable, non-enveloped. And then people studied it from a clinical point of view, but they also realized that it was a very good model system to study virology because it was stable, grew well in tissue culture.
Right.
You could isolate it on cesium chloride gradients very easily, so you could purify it to homogeneity.
It's easy to draw a cartoon of it.
It's a very easy to. It looked like Sputnik, so Sputnik was in it was 1958 or 9. So this thing, it's an icosahedral. It's a sort of classic icosahedral symmetry.
Which was, of course, discussed last week.
Fibers sticking out of each of the vertices. So the not the archival. What's the word I'm looking for? The archetypal picture was taken by, I think, Richard Valentine as an EM. It was also coincided with a big upsurge in EM work on viruses and what they looked like. And of course, here was this gorgeous icosahedron with the fibers sticking out of each of the vertices, and he was able to capture that on an EM. On an electron micrograph, and it just looked so gorgeous. It just looked like everybody's idea of how a virus should be. It had these little stalks sticking out which would attach to cell surfaces.
Sure.
The irony, of course, is that it took a very long time to work out what the receptor for the virus was. Unlike other viruses, it took much longer. But you could see immediately how this thing would attach to cells via this external
So why
appendage.
is it limited to recruits, military recruits, or is it infected?
No, it's more common in recruits than, say, if I remember correctly, than, say, university dorms. But I think that's partly a question of crowding and the conditions under which recruits find themselves. So I think it was also found in institutions. For when people when people were institutionalized, like jails. It's not so much jails, but things like orphanages.
Oh, I see.
Where you had large numbers of susceptibles crowded together.
Gotcha.
And it's not limited to the United States. There were big outbreaks in Holland, for example, which were examined by the Dutch army. So it was not restricted, and of course it's widespread in the general population. And then they started to discover that there were many of them. There wasn't just one adenovirus. There were lots of different serotypes.
How many there? 50 some odd?
The last time I remember there was 41 or 2, something like that. But there may be more now.
But the one everybody worked with was Adeno 12, as I recall.
No,
I don't recall that.
Well, many people did work on adenovirus 12 because it was discovered to be capable of transforming.
That's why they worked on it.
So they worked. It was a transforming virus.
Got
But the initial isolates were 1, 2, 3, 4, 5, and 6.
Sure.
And groups worked on adenovirus 2 and adenovirus 5.
I see.
51. 51.
51 now. Okay,
varieties.
51.
Dick, excuse me. Dick
I'm sorry,
has to-
I do have to leave. I have another appointment I've got to keep, but I would like to mention my book pick of the week.
Please.
It's a softcover book. It's called The World's Water, 2008 and 2009. It's a biennial report on freshwater resources, produced by the Biennial Report on Freshwater Resources Committee, and it's produced by Island Press, and it's everything you ever wanted to know about why people are going thirsty throughout the world.
Right.
Which, of course,
West of the
relates very
Mississippi
much.
is going to be very true.
Water is the new oil. That's what they say. And then, we're having a roundtable discussion of this tonight as part of the World Science Festival, which I am a speaker of. So I've got to bring this book with me to tell them, yep, I've done my homework. See, I've read this whole book, and that's not true at all, by the way. I just got it last week. At any rate, I'm sorry to have to leave the discussion. But
All right, thank you, Dick, and we'll see you in July.
You got it.
So apparently this is responsible for about 10 to 15% of respiratory infections?
Yes. There was a very large surveillance study, I think, in Seattle, as I recall, where they... it may have extended beyond Seattle, but there was a very large surveillance where they tried to work out what upper respiratory diseases were caused by, and adenovirus was in sort of 5 to 10% of standard colds.
And it also causes some gastrointestinal
yes. Oh, yes.
disease,
So it turns out that adenovirus doesn't only cause upper respiratory tract infections. There is a very interesting serotype specificity to the diseases they cause. So serotype 8 causes epidemic keratoconjunctivitis, which was first discovered, it was called shipyard eye, because I think...
I love that name.
Shipyard eye. And the reason I remember Harry Ginsberg telling us about this, it was an eye wash station which was in a shipyard where they were building ships, I think for World War II, but it was certainly at a time when America actually built ships, so it's some time ago. And with the iron workers would get shavings from metal working into their eyes, metal dust essentially into their eyes, and to take care of that, they would go to the eye wash station and wash their eyes out. Well, somebody had a case of adenovirus type 8 and had keratoconjunctivitis, and this spread. There was a case cluster in this particular shipyard. I don't know, Bethlehem Steel or something in Philadelphia. I don't remember where the shipyard was, but and it turned out that this was a specific adenovirus serotype 8. I've got personal experience of this because some years ago my wife came down with a bad case of keratoconjunctivitis, and I was petrified that I'd brought one of my recombinant viruses back from the lab, so I insisted that they serotype it.
Yeah.
And the lab very courteously did so and said, no, it's standard type 8, which relieved me greatly. But the interesting point of this story is not my anecdote, but it turned out that her ophthalmologist's office had several cases of type 8 keratoconjunctivitis. And almost certainly the source of infection was the machine used to measure eye pressure or to measure one of the things that ophthalmologists' office do. It's very difficult to sterilize those between patient visits. So next time you go to the ophthalmologist, make sure they're sort of swabbing things down. I mean, not that an alcohol swab will do very much.
So it's quite a stable virus.
It's a stable virus. So it's not killing the virus. It's just washing it away, essentially.
So why don't ophthalmologists' offices have that happening more often?
I think because it's not a particularly common epidemic. It has clusters, and that's why it's called epidemic, because you get these large clusters of cases. I don't know why, unless their hygiene is particularly good, so it doesn't happen very often. I don't really know the answer to your question, Alan.
Okay.
It's certainly not uncommon to find a cluster which is associated with a particular ophthalmologist's office.
Would this be the same serotype you would be infected with in a swimming pool or some kind of...
Well, I think that's another source. That's another source of infection.
Eye infections, yeah. I've heard that as well. Improperly sanitized pools and hot tubs, that sort of thing. Yeah.
I don't know how susceptible it is to the levels of chlorine that are put into that's something I don't know.
Yeah, I think when you don't chlorinate properly,
yes, right. Now then, so you were asking about other serotypes. So serotype 40 and 41, which is why I remembered 41, cause gastrointestinal disease in newborns or early infants, and that's a serious problem in postnatal nursery settings. So it can spread through. In fact, sometimes causes neonatal units to be shut down because it's very contagious. And of course it's transmitted fecal-oral route, so if it's causing gastroenteritis among newborns, you can imagine what a problem that is. So, and it has a high morbidity. I don't know what its mortality is offhand, but it's something serious. They have to be careful about.
Have people developed vaccines against particular serotypes?
Yes. So there is now vaccines against the acute respiratory disease of recruits. And of course, what happened, this is a classic example of a vaccine being effective against a particular serotype. I don't remember which one. Let's say it was type 3. And then I think it was actually type 7, but whichever type it was, then you have an outbreak of a different serotype, which the vaccine doesn't work against. So, yes, there are vaccines which are administered as far as I'm aware, were only ever administered in the military. And I think that the one I'm aware of was adenovirus type 7. I don't think the military does that anymore.
But
Now,
right, it didn't wipe out the disease. In other words, it just changed its ecology.
Yeah.
So they made a vaccine against types 4 and 7.
Yep.
And they were used from 1971 to 1999.
Right.
And then they stopped.
Right, they stopped. And I don't know, I'm unaware of whether there has been an upsurge in acute respiratory disease of recruits, or whether the living conditions of the military are different and better than they were.
I was on study section last week, and there was a grant on adenovirus type 14. So I listened carefully, and it turned out that there was an outbreak of this in 2006, 2007 in 4 different states. And they were not military people, they were civilians. And so this was not a serotype that was seen very often.
Right.
And so they said, well, maybe this is going to cause a global epidemic, but then it was not seen again after that. So I think they pop up now and then, different serotypes, but we don't know what happens in the interim. We don't even know if there's a vector or animal host for these, do we?
I think there's not an animal host.
It's all human.
No, it's all human-to-human transmission.
Are there animal adenoviruses, though?
Oh, there are lots of animal adenoviruses. They come in 3 broad flavors. I actually worked a little bit on this. There's what are called mast adenoviruses, which is, as its name implies, found in mammals. There's aviadenoviruses, which are found in birds and some reptiles. And there's a group which is called adenovirus, AT adenovirus, because they have a very high AT content, not because of their host predilection. And those are found in mammals, birds, and reptiles. I think I'm right in saying that. It's a long time since I've looked into this. I'm a bit hazy on this. But they are quite clearly distinct evolutionary trees. They can be separated quite easily on the basis of sequence. But there's a whole cluster within each lineage which are similar. So, for example, it's thought that one of the bovine adenoviruses, there's a group in Hungary who's done a lot of interesting work on this. Some of the bovine adenoviruses, most bovine adenoviruses are mast adenoviruses. They're adenoviruses of mammals. But there are some which are clearly closely related to avian adenoviruses. So the assumption is that they crossed over when agriculture allowed both. It's the old mixing vessel story, only slightly different. When you had avian species and bovine species together, there was a transmission from one host to the other. So the evolutionary history is quite interesting. We were interested in it because we wanted to look at the structure of the adenovirus major late promoter. So we took a whole series of promoters from a variety of species, and one of my students sequenced the surrounding promoter area, and you could clearly see that there were different lineages even then.
So I guess there isn't sufficient reason to have a vaccine in the general population.
No, there isn't. I think it's a problem which is akin to the problem of rhinovirus, that you'd have to make a large number of different serotypes. You'd have to make a vaccine against large number. Now, it's not 105 or whatever it is in rhinovirus, but it would be certainly dozens.
Still 50 some odd total, 51.
Yeah. Well, you wouldn't have to make it against 51 but you do it against the common one. So maybe 1 through 6, 7 or something like that. I don't remember what the ones that cause the upper respiratory tract infection.
So why isn't adenovirus as successful as influenza?
Well, I suspect it's the mutational rate.
Yeah.
I mean, I suspect it's a simple, and I would dispute the business about being as successful because
it's still here, isn't it?
It's still here, and it affects. If you think 10%, let's say 10% is the number of seasonal colds, or not seasonal, but upper respiratory tract infections, 10% of them are caused by adenovirus. It's doing very nicely, thank you. Its host is living, spreading it to other hosts. You have long-lived immunity to a particular serotype, but there's plenty of susceptibles coming along.
So I guess in general it's less severe than influenza.
Yes,
and the fatality rate is lower,
very low.
Much lower than the 0.5%.
Yes. That's my understanding. You can get a pneumonia. There are cases of pneumonia. I don't remember if they're in, again, people who have some compromise. That I don't remember. But it's not common. I mean, same way with rhinovirus. You don't. It's largely upper respiratory tract.
Now, when adenoviruses have been used as gene therapy vectors, which is, I think Vince was going to get to this in a moment. Right. I see it on the agenda, but you're talking about pathogenesis.
Right.
One of the obviously the big dust up a few years ago was that a patient died in a gene therapy trial. Jesse Gelsinger died in a gene therapy trial after receiving a dose of a genetically modified adenovirus.
Yes.
What does that? I mean, I remember reading the story at the time, but I've gotten murky now on the details of what happened with that particular patient. What was the response there compared to the regular response to an adenovirus?
Oh, I think it was. My guess is that it was an overwhelming immune response to a very large dose of purified virus. This was to treat, if I remember correctly, I think it was Gaucher's disease.
I think that's right.
And the virus will, when it's inoculated intravenously, which I think it was, will travel to the liver. And as I recall, the patient died of an overwhelming probably innate immune response to this huge dose of antigen being administered. That I think is the case. Mm-hmm. I could be wrong, so I don't want to be held responsible if I've got it incorrect. But it was not a normal response to the kind of infection that you would get in the upper respiratory tract. These viruses were designed to be, and essentially are, non-replicative. Now, that has to be couched a little bit, because in fact, there is a little bit of replication that can occur with these vectors, which is one of the issues that people had to try to address. But broadly speaking, this virus was not replicating like a normal adenovirus would. It was its initial early genes were removed so that it basically couldn't replicate, and it had a vector in it. So it had whatever the gene was. I guess the replacement gene for Gaucher.
Actually, I just checked this on Wikipedia, and it was an ornithine transcarbamylase deficiency.
Okay, ornithine transcarbamylase deficiency. Okay, so that gene was the gene for that enzyme was incorporated into it. So the response, it was very atypical for what happens in a normal response. So you get an upper respiratory tract infection, you get a standard immune response with eventually, I guess, IgG2 being made against the virus, and then you have lasting immunity. So this was giving, as I recall, between 10 to the 8 and 10 to the 9 virus particles in an inoculum. Huge amount of inoculum. I don't think this response was ever seen in any other patient. So there may have been some particular issue with this particular patient as to why he mounted that particular response. But it did give people pause, needless to say, that this approach had severe problems, which had not been fully anticipated.
Have we resumed using adenoviruses since then? I think so, right?
Yes, I mean there are I think clinical trials still going on with some adenovirus vectors, but the initial hope has not been fulfilled.
Why were they initially thought to be so useful for gene therapy?
I think it's a historical accident to some extent, that it's a virus where the genetics was very easily manipulated, where it was very easy to make recombinant adenoviruses. It was very easy to make, in the tissue culture situation, it was easy to make vectors which would make large amounts of the protein that you were interested in. It had a big carrying capacity. You could remove quite a bit of it so that it became non-replicative, and you could stuff all kinds of... So, for example, if you wanted to put muscular dystrophy, the dystrophin gene in, you could sort of do this because you could take... Well, I can't remember the size, but you could put in a whole chunk of it, at least, by getting rid of most of the adenovirus.
Probably we should tell people what The DNA, the genome is
So
DNA.
it's a DNA-containing
Double-stranded.
genome. Double-stranded. The ones that are widely used have about 36 kilobases of double-stranded DNA. You can get rid of a large proportion of that and put in genes. When I was working with it, we would routinely put in 4 or 5 kilobases of DNA, which was enough to put a therapeutic gene plus controlling elements and so on. Subsequently, people developed methods to basically, there was a wonderful term, they called them gutless adenoviruses, which Frank Graham, who was one of the major players in this field, to whom a lot of credit should be given, Frank Graham objected very strongly to that terminology. He said it should be called gutted adenovirus rather than gutless, because gutless sounded as though they were useless. And eventually people found ways to make adenoviruses which basically had the ends of adenovirus and everything else was the gene of, or genes of choice. And so there are ways of making those viruses. So they had manipulative advantages.
Plus, they didn't cause such serious disease.
Secondly, they didn't cause serious disease. But of course, the viruses that were made shouldn't have caused disease at all, actually.
Yeah,
so that, in a sense, that was a sort of moot point. And I think the carrying capacity was one of the things that really intrigued people. You see, the other kinds of DNA vectors like AAV have a limited carrying capacity. You can't put a lot of stuff into an AAV genome and still stuff it into the viral particle, because the viral particle's small. And it can only carry, 3 or 4 kilobases of DNA.
Sure.
So you're kind of stuck there a little bit.
What about the fact that there are a lot of serotypes? Was that useful?
Well, that was another thought was, of course, that you could switch serotypes. So if somebody was immune to, say, type 2 but not type 5, you could then use type 5. Or if they became immune to the thing that you were giving them, you could then switch to, well, this is all pie in the sky. You can't tailor medicine that way. You can't say, well, sorry guy, yours didn't work. We'll just go and make an adenovirus 12 vector. I don't think that's a realistic goal.
Right. Hold the elevator. I'll pop over to the lab and do
that. Yeah, that's right. I'm just going to tailor up another one for you. But there was a feeling that people might not be, there were a variety of serotypes out there, and you weren't probably immune to all of them. So you could get some way of doing that. The other thought, I think, was that should there be any escape of this vector, if this vector should replicate, you were likely to mount a pretty solid immune response to it. So you could eliminate it if that became a problem. Right. So there was a period when people thought it could be used as a gene therapy vector. That is specifically for gene therapy. So you replaced a gene. Now, the problem with that is quite obvious. You're going to clear this virus. This virus does not integrate into the human genome. So you'd have to keep on administering this because the protein is made and you cure cystic fibrosis for a month, but then you've got to deliver it again. So there was no way to integrate the virus into the genome, at least in anything other than sort of chalkboards on chalk on
So in a normal replicative cycle, the virus DNA never integrates.
Never integrates. No, it goes. Now, in experimental circumstances, adenovirus will transform cells, and there was a big effort in the 70s and 80s to see whether any human tumors were caused by integrative versions of adenovirus. Maurice Green did a very large study on this, and basically no evidence at all for any association between any human tumor and any adenovirus. So that worry, I think, wasn't realistic, but the problem is if it doesn't integrate, then of course you've got this, are you going to continually have to give therapeutic doses of this virus? And of course you're going to make an immune response, and so it's going to get less and less effective. So then the other area that people thought this might be very valuable was in tumor circumstances. So the virus didn't have to replicate very long. You could design a virus which would zap the tumor that was troubling you. And there was initially some quite interesting seemed to be effective treatments for head and neck tumors, that you would inject the virus directly into the head and neck tumor. And I remember going to meetings where it looked as though it was efficacious. And this is a very difficult tumor to deal with. surgically difficult to remove, very painful. It was a good candidate for this. But that clinical trial failed. It was not effective. And as I recall, they always gave cisplatin in addition to the virus because they were, it was unethical not to do so. So it never, as far as I'm aware, ever panned out. So from a therapeutic vector standpoint, I think the days of adenovirus vectorology are over. Now, there are many other circumstances where the virus is useful, certainly in experimental circumstances, having vectors that will take some gene of choice to some part of an animal.
We use them.
And you use, Vince knows all about this. But from a therapeutic standpoint, I think it's...
Well, let me tell you what I see. I'm on the biohazard committee here. We have a lot of protocols using adenovirus for immunization, especially against HIV.
Right. Now, that was the third of the triad, right? So the last great hope is that the virus, since it is immunogenic and since it does replicate a bit, and you can put large amounts in, you might be able to use it as a vector for immunizing. Now, those things keep on going. I don't know if any of those have become valuable. I know that there was a great hope for an HIV vaccine. NIH put a lot of money into a company called Genvec, with which I used to collaborate. And I think they had a clinical trial with Merck or one of the big...
There are several, and now the protocols mainly involve first giving a DNA vaccine, and then boosting with a adenovirus vectored protein.
And is that working? I haven't said
nothing really jumping, but they're continuing. They just try.
Well, I think it's the one area that I would think would have some usefulness.
And there are anti-tumor approaches where you put various cytokines in this way and so forth, but nothing is really...
Nothing has really...
Not yet. But you have to to try, as you know. These are good. Yeah. Maybe you could give us just a little primer on the biology of the virus and how it works in a cell, and what are the big things that we've learned from studying adenovirus? I think there were a few.
Yes. Splicing, to name but one.
That's right.
The virus is a DNA-containing virus. It attaches to a cellular receptor, which is now known. It's called the Coxsackie adenovirus receptor because it's shared with Coxsackie virus. This is an integral membrane protein. The virus attaches. It's brought in by receptor-mediated endocytosis. It makes its way to the nucleus. The DNA is then transcribed in a classical three-step process: immediate early, and late phases.
This is by host enzymes.
By host RNA polymerase II. Some of the early factors that are made are transcription factors, which modify the behavior of both cellular genes and viral genes. The next phase is characterized by a very rapid ongoing DNA replication, which is programmed by the genes of the delayed early, if you like, set of family of genes. Then it goes into the late phase when it makes the structural proteins, large number of different structural proteins, which are formed into the icosahedral structure, which is formed in the nucleus. The virus remains cell-associated to a large degree. So you have to find ways of doing a plaque assay, which of course requires cell-to-cell spread. So you need to get the virus artificially, in a sense, from cell to cell, and it does lyse the cell and move from cell to cell.
So the DNA polymerase is a virus enzyme.
It's a virus enzyme, yes. So it was not, initially it was thought to be a good model for DNA replication. It turns out to be a very interesting DNA replication cycle. It's a displaced single strand, which is displaced, then forms a panhandle, a looped panhandle, which is then replicated back to complete the duplex. So you make two duplexes in a sort of staggered temporal way. You end up with two duplexes from one in the classical Watson-Crick manner, but it's done in a staggered temporal way.
So it's different from a host.
It's different
DNA
from the host,
and
it has a DNA binding protein, a terminal protein which is covalently attached to the ends of the DNA molecules, which is integral in the initiation reaction, and a polymerase, which is specific for the adenovirus.
So how long does the whole cycle take, roughly?
The DNA the whole infectious cycle. The infectious cycle is quite long. You can start to see release of infectious virus or the production of infectious virus, not release of production of infectious virus after about 15 to 18 hours. So it's a fairly lengthy. It's not like a polio.
Polio's 8 hours.
Yes, right. So the whole cycle in a typical one-step growth curve would be over between 36 and 48 hours, depending on tissue culture conditions. So it's a fairly slow process.
It's quite slow, yeah. So
now, you asked what
have you learned?
Yeah, so what have we learned? Well, so it was integral in understanding how DNA-dependent RNA polymerase makes RNA. It was used to show that RNA pol2 is the transcribing polymerase. All of the RNAs that are made, almost all of the RNAs of the virus are made using RNA polymerase 2. There's one exception. RNA polymerase 3 makes two little RNA molecules called VARNA. Yeah. So, and then of course the big discovery was that the virus is transcribed in the late phase of infection to make a very large transcript, which is then spliced to make sub-transcript-sized mRNAs. So it was used extensively to study RNA transcription.
So who made this splicing discovery?
Phil Sharp and...
I've heard of him.
And what's the
Rich
other?
Roberts.
Rich Roberts, right? Cold Spring Harbor.
This was totally unexpected, I remember.
Oh yes, it was. I mean, I remember Harry Ginsberg coming in and saying he just had to talk with somebody who'd been at the Cold Spring Harbor meeting of '77, and we just, our jaws just dropped because we'd always been brought up on the one gene, one enzyme, one gene, one RNA, one enzyme hypothesis, and here was something which was completely unexpected. This was certainly unexpected by me.
And of course, then we found that our genes, in fact, are spliced.
Yeah, right. So then,
introns.
in very short order, SV40 was shown to be the same way.
Yeah.
Many viruses turned out to be. And then, of course, the host cells.
And a good part of our DNA is intron.
Yeah,
these
exactly.
spliced out parts.
Now, adenovirus doesn't have very much that's not used for transcriptional, for encoding purposes. Very little turns out to be that way, but of course a virus pares it down to the minimum.
But it does this to get flexibility in what it can code for.
Exactly.
Right?
So you can code for a bunch of things from one transcript, which turns out, and you can use different reading frames. And there's a whole, any particular permutation that you care to think of can be found, I think, in adenovirus. The one other thing, of course, is that this turned out to be a fertile field for studying transcription factor, too, because the promoters of the virus were widely used to isolate and characterize cellular transcription factors, which were DNA binding proteins which affected gene expression. So that was, if you went to meetings of adenovirus and SV40 in the 1980s, this is all you heard, was all about transcription, particularly about the transcriptional control, because the virus had these promoters that were clearly being controlled in a temporal fashion. How did they do this? Well, they had transcription factor binding sites and so on.
Is that because we had viral DNA that we could use as a template early on before we had a cellular equivalent?
I think it must have been. I mean, it was an easy thing to use. It was always touted as being the sort of the window into how the cell worked.
Sure.
And of course, since it used RNA pol2, this was the way it would be. Right?
Yeah.
And then it turns out there are lots, subsequently, every half decade or decade, some other really interesting thing would turn up using adenovirus. Modulation of the immune system. One of the first areas where this became really clear was with adenovirus. Bill Wold was studying a region of the virus which was called the non-essential region, much to Harry Ginsberg's annoyance, because he said it's not non-essential to the virus, which of course was true. The virus has got all these genes, which turned out to modulate the immune response, either by affecting class 1 MHC molecules or you name it. There were ways to. So there was a cassette of genes which did stuff. I got interested in genes which modified the DNA damage response. And these were coded by genes in what was called E4, which is the extreme right-hand end of the genome. And they turned out to modify how cells respond to DNA damage, because the cell sees this piece of DNA floating around in a sort of You can think of it as just being a bit of rogue DNA. So the cell says, "Hey, what's going on here, guys?" and has to deal with this, and doesn't, because the virus subverts the DNA damage response.
I know also that a big area of work is the structure, because it happens to be a very large virus.
Yes, right.
And initially that was a challenge.
Right.
You know, the first structures, the X-ray structures, were done with small viruses like polio, plant viruses.
Right.
And with adenovirus, they started by crystallizing components.
Components, yes.
The hexon, the fiber.
Right.
And then they would make a cryostructure of the virus and fit these bits in. In fact, there was someone here who...
Roger Burnett.
Who was early on here, and then he moved to Philadelphia.
That's right. No, Roger Burnett actually did some of the very earliest structural work on the virus, which turned out to be very interesting indeed. Because it's a big virus, and you can crystallize it, I think, but I'm not sure the crystals are any good. But it's you can I think you can crystallize the virus.
No, I think that set the way for the herpes structure and others. It was really groundbreaking.
It was bringing the resolution from two ends, from the sort of atomic end with crystallization and the optical end with all of these EM and associated techniques, and it was fascinating to watch that develop.
Yeah, it's beautiful stuff. So do you miss working on adenovirus?
Oh, yes, I miss it greatly, but I don't miss anything else.
You don't miss writing grants?
No. I never minded writing grants. I mean, even if I didn't get them.
You would not like it now because that's very frustrating.
But what I mean is that the actual writing of the grant is fine. It's all the rest of it.
Well, hey, when you have plenty of ideas and results, writing a grant isn't difficult. It's when you don't have either.
Well, it's when you get rejected and
over and over. And that's what happens today. You get rejected. You have to be used to
I retired. I was very fortunate because I retired just as the expansion period of NIH stopped. And then I would come back and I would hear my former colleagues saying, complaining. And I could tell that I would have spent what have been my retirement years just sitting in my office writing and not getting funded. And it would have been very frustrating.
Well, instead you can come here and talk with us about viruses. Well, that's adenoviruses. That's great. I really like that. I think adenos are always interesting. So let's do a couple of emails and then wrap things up here. We always get a lot of emails. It's great. Jason wrote, a quick look at Wikipedia shows 3 animals that were domesticated from Africa: the donkey, sub-Saharan cow, and the guinea fowl.
We screwed up. Yes.
So a couple of weeks ago, Dick quoted Jared Diamond as saying no African mammal had been domesticated. Yeah. That was his statement, right, Alan?
Yes, that is what he said, and I'm not sure that Jared Diamond actually said that.
I don't think he did. I don't think he did.
Well, Dick insists that Jared did, but I'm not sure. But anyway, we were wrong, and thank you for correcting us, Jason. And that's why this is not This Week in Mammals. It's This Week in Virology.
Well, we get corrected on that, too. So.
Yeah, you're right. I don't mind. Well, the internet is like science. It's self-correcting.
Right.
Brian wrote, my son is away at college this summer doing research. His roommate just arrived and was sick. The roommate went to the clinic on Monday, diagnosed with swine flu, and immediately sent back home. Since then, 6 different agencies have called my son and offered to give him Tamiflu, but he's refused. What would be the point? I'm not aware that Tamiflu would prevent an infection. Would Tamiflu reduce the potential spread by reducing the time my son would be shedding virus? So far, my son seems to be fine. My son is an EMT and volunteers with a couple of rescue squads. He has decided not to show up for duty for the next 10 days to avoid any notification headaches that he might give government agencies if he does get sick. Thanks. I enjoy TWiV greatly. Tamiflu it can be given prophylactically, actually. Typically, it is given within 2 days, I think, of infection, and you have to take 2 doses a day for 5 days. And if you take it within 2 days of symptoms, it can lessen the severity, and it will also reduce spread because it reduces virus production. And I looked up on the CDC site their recommendations for prophylaxis. This would be for healthcare workers and anyone in contact with others. And prophylactic doses are recommended once daily for 10 days following close contact with an infected person. And that should happen within 2 days of exposure. So people who are working with others, like your son, an EMT, it's not a bad idea to do it within 2 days. Now it's too late, of course, so he shouldn't bother. There's no point in taking it if he doesn't have any known contact with anyone else. But it is effective at reducing disease severity and spread. Of course, if the virus is sensitive, as the one is now, but if it's resistant, then it's useless. So the H1N1 is sensitive so far. So I'm not giving advice, but that's the science.
All right.
Richard wrote, I'm still finding your podcast interesting. Oh, I hope so.
Good.
After all, a day where you learn nothing is a sad day indeed. I do a lot of driving in my job, so podcasts are an ideal medium for me to learn. They don't get in the way of driving. If anything, it helps me to keep alert as my mind stays active. I drive over 1,200 miles a week, so I have quite a bit of time to listen.
That is a lot of driving.
I'm sorry. I have a few requests for topics, although I am in no hurry. You seem to be able to find plenty of material to cover. However, here is a short list of things that interest me. So we'll do these probably in future episodes. How does the virus...
That's an excellent list of topics,
get
actually.
into a cell? Is it different for different types of virus? How does the virus repurpose the cell's machinery?
Oh, these are good topics.
How do different viri, or viruses, we say viruses, move from the infected cell so as to be released into the environment? Are there any novel tricks used? Sure. So we're going to talk about all of these.
Yes.
Thanks for the coverage of flu. It has been great to have a counterpoint to the media hype.
And by the way, Richard, you have just hit on 3 of the biggest questions in virology. That's,
yes.
Off the top of your head, so
Right.
that's pretty good.
That's pretty good. You're doing well.
No, it's often people who don't study viruses that come up with the
Lateral
questions.
thinking.
It's
right?
good. So you've been paying attention. That's great.
Yes.
So he has a book to recommend to us, possibly ideal for the less scientific listener or those who are specialized and would like a broad view of what has been discovered by us humans. The book is A Short History of Nearly Everything by Bill Bryson.
Yes, that's a good book.
Yeah?
Yeah.
He's listened to it on audible.co.uk, and he thinks it's available on audible.com.
It is. It is.
Are you an Audible listener?
Rephrase that. It's certainly available as an audio book, that I know, because we gave it to my brother-in-law.
Okay, great. Many thanks for the time that you put into your podcast. So actually, Dick and I are recording some short bits on basic virology, and we're going to cover a lot of these steps in the future episodes. And finally, one more. Raphael wrote. So Raphael is an MD from the Philippines. I am Dr. Raphael, a practicing physician in Cebu, Philippines, a recent TWiV listener and one of your Twitter followers. I happened to come to your virology blog when I was searching for resources regarding the recent H1N1 flu outbreak, which I will be talking about to a group of lay people in a company where I hold my clinics. Aside from that, I teach topics on medical care for mariners and seamen, particularly on the subject of communicable diseases and sexually transmitted diseases. I also do part-time transcription work for an American company based on the East Coast, for which I do receive compensation, but I also transcribe parts of the Medicast podcast for James Davis for free, since I also learn much from his shows, which leads me to the reason why I sent this email. It's because of the discussion you had with Dr. Palese almost a month ago. I learned much from that one single interview that I think would be helpful for me in educating my patients and friends whenever they ask me something about the new H1N1 virus. Although that was the time that the outbreak was still unfolding, I think listening to your later TWiV podcasts would unveil other important facts that would answer some of the questions or update some of the concepts that both you and Dr. Palese raised. The problem was that there were no transcripts on that episode and the episodes after, so seeing the need, because I sometimes am more comfortable reading than listening, I took the liberty of transcribing first the episode with Dr. Palese, while the others would be later, or until you would be able to get somebody else or some company to transcribe. I hope you can share the attached transcript to my other fellow TWiV listeners who may also be interested in reading this special episode.
Awesome.
So he sent a Word document, which I posted on twiv.tv. So we thank you very much for that. That's great.
Nice.
I'm trying to get some transcripts done. There is a company in the UK that does it. I think it's called Pods and Press, but they charge, and it's $1 a minute. Oh. And since we're over an hour, it's too much. And they would do it for free, but then they keep it, and you have to subscribe to their service to be able to see them. And I would rather have everyone have access to them. Right. So if there are others who would be interested in transcribing, I know for other podcasts people volunteer to transcribe. That would be great. I'd love to hear from you. So let us know. Maybe we could work out a deal, and maybe you could get a copy of my textbook for X number of transcripts or something like that, so if you're interested. And that should do it for emails for today. And finally, our science picks of the week. So Hamish, we've come to picking science things, for each of our participants now pick something science, a book, a website, anything. And Alan and I and Dick picked that water book.
Right.
And Alan and I will give some, and then maybe you can think of something that you like to share with our listeners in the meanwhile. So, Alan, why don't you go ahead first with your pick?
Sure. So my pick this week is a website called Eureka Alert, which I refer to pretty much every day. I get the RSS feed from there. I also get some emails from them. And Eureka Alert is a service that was set up by Science Magazine, but it's a press release service. It's freely accessible. And these are press releases that are sent from research journals, from universities, from individual researchers, in some cases from companies, but mostly it's aimed at the basic research side. And they are press releases about new research that's coming out. This is one of the main sources of finding new stories. Is browsing through the day's press releases. Of course, if you're an actual reporter, you then have to dig deeper to get the real story behind the press release, but the releases are often a good starting point so that you know what's going on. And you can go to Eureka Alert. Anybody can go there and read the publicly available releases. They also, if you're a credentialed reporter, they have the embargoed press releases about papers that have not come out yet or not been made public yet. But it's grouped by subject, so you can choose medicine and health if you like, or agriculture, or archaeology, or technology, and you visit a topic and you get everything that's come out today. You can go back through the archives. If you do read this in an RSS feed or decide to subscribe to their emails, you have to watch out for the information overload that you're going to get, because a lot of people don't understand how many thousands of these types of releases are produced in a given day. But it is, if you don't mind drinking from a fire hose, it is a source of some very interesting information.
So this is put out by AAAS, right?
Yes.
Great. So this is how you get a lot of your good stuff for TWiV even, right?
Yes, I've picked a couple of things out of there.
So how do you get to be credentialed reporter? What do you do? You have an ID or something?
Well, there are different meanings of that. If you're in Washington D.C. and you need credentials, it means you have to go through a government agency in order to get clearance to go into a press conference. But for a service like Eureka Alert, they have embargoed press releases. This is about a week before an issue of Science or Nature or some other journal comes out. They'll send out information about what's in that issue to reporters, so that reporters can research the stories and have them ready to go as soon as the paper is actually published.
Right. So are you credentialed?
I am. And
can you show them to us right now?
I can't show you press releases that are embargoed.
No, credentials. I want to see.
Oh, my credentials. I've got my credentials right here.
Is it like a physical thing? Is it a piece of paper?
No. I don't. It's not. I'm not given a card or taught a secret.
I hope you could show us a card. All right.
No, sorry about that. But yeah, there are a couple of mechanisms for doing this if you're in the business.
Okay, so that's Eureka Alert. Thank you. And Dick, of course, picked The World's Water by Island Press. My pick is a blog called Respectful Insolence. And this is a blog by, well, I don't know who he is because he doesn't use his real name, but he is a surgeon scientist with an ego big enough to delude himself that someone is interested in what he writes. And he writes about science, and he has a nice quote here: "A statement of fact cannot be insolent." So it's called Respectful Insolence. And I've been reading it lately because he's been posting a lot about swine influenza. And he has posts like "Academia Slowing Down the Search for Cures," the AP shoots and scores again, "Holocaust Denial and Hate," "Patient Empowerment," "Who Should Call the Shots." Check it out, Respectful Insolence. So, Hamish, you got anything to share with our listeners?
Well, I don't keep as watchful an eye as either of any of the participants, of course, because I'm retired. But I would suggest, since it came up earlier in the discussion, If people have not read Guns, Germs and Steel, they certainly should read that, because I think it's a very interesting, very provocative book with lots of lessons to be learned as we proceed in various epidemiological circumstances. The other book that I read, and this is not, again, infectious diseases, but a book that I found really very interesting, was a collection of writings by Atul Gawande, who writes, I think, for The New Yorker.
Yes.
About his experiences as a surgeon and trips to India, dealing with how India deals with medical situations and how impressed he was. The reason I raise that is I think that we're facing a crisis in this country, which has been raised a number of times, with healthcare provision and so on and so forth, and what are the society responses to healthcare and what are the ethical things and so on. And I think he writes extremely well, very cogently, about dilemmas that doctors face, both in the first world and India, which I guess is in the sort of 1.5 world now. I don't know what one would call it. So I found that a very interesting book, and I just can't remember the title, since I didn't expect to be asked, but it's by Atul Gawande. Yeah. Maybe Alan will find it for us.
Working on
He's
it
working
now.
On it. In the meantime, thank you for those suggestions, Hamish. That's great. And don't forget, you can find other science podcasts at sciencepodcasters.org and promednetwork.com. We're members of both networks. Alan, have you got any info on that thing?
I've got his latest book is called Better.
Mm-hmm.
I think it's the one before that was the one I read. That isn't ringing a bell.
Complications. A surgeon's notes on an imperfect science.
Yes, that's it. Yes.
Complications.
Yes. Complications. A surgeon's notes on an imperfect science. That sounds like the title. It was a collection of his New Yorker writings. I suspect Better is also, either of them would probably do, because he's got a particular flavor to his viewpoints, and I think this audience might well enjoy them.
Very
and
good.
He does deal a little bit with infectious disease, but it's mostly other forms of medicine.
Great. Well, thanks very much. And thanks for joining us again,
Oh, yeah,
Hamish.
I loved it.
Appreciate
it was great.
it.
Yeah.
And I understand you're heading off to Nova Scotia for the summer.
Yep.
Well, we envy you.
Yeah.
Have a good summer, and we'll have you back in the fall.
Excellent. Love to come back.
Alan, thank you again.
Always a pleasure.
Good to talk to you, Alan.
Yes, good talking to you.
Yep.
Alan's at dovedocs.org.
.com.
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 I'm going to get it right one of these days. dovedocs.com, and Alan Dove on Twitter. You can follow TWiV at PROFVRR. I'm also on FriendFeed, friendfeed.com, PROFVRR. And we have a website for TWiV, twiv.tv. We have show notes, links to articles that we talk about, and of course you can always download the episodes there as well as on iTunes. So keep sending us your comments and questions, twiv at twiv.tv. You can send us an audio file if you'd like as well, an MP3. Thanks for listening. We'll be back next week. We have another guest next week. You've been listening to This Week in Virology, a podcast about all kinds of viruses that have nucleic acid genomes. How's that?
That's good.
That's it. Yeah.
Thanks for joining us. We'll be back next week. Another TWiV is viral.